Today's presentation is a review of research and experimental design principles with a focus on how we approach primary research in science and engineering. We'll cover key elements of the scientific method, how to formulate questions and hypotheses, and the components of a strong experimental design. Let's dive in. So just as a reminder, when we think about primary research includes original investigation, so studying something that's brand new, adding to the scientific knowledge that's already out there, collecting data firsthand, so new data that's being collected, and also going through and publishing in a peer-reviewed journal. So taking your study and putting that out there to the scientific community. The scientific method, as we see here in this diagram that was created by Amy Cohen for Science Buddies, starts with asking a question. And then we do background research. And from our question, our observations, and our background research, we form a hypothesis. And from that, we create an experiment to test that hypothesis. Once we've been working through the experiment, we want to make sure that the procedure that we're doing is working well. So evaluate the experimental quality. If the answer is no, then we want to troubleshoot the experiment to make sure that all steps in the setup are going to be working properly to test our hypothesis, and that the data we're correcting are precise and accurate. If it is working well, then great, we continue. We can keep collecting those data and then eventually analyze those data. And then from that, draw conclusions. From those conclusions, this is where we go through and start aligning the results and our conclusions to the hypothesis. Does it support the hypothesis, or do we reject the hypothesis based on our results? Depending on the outcome, then maybe we create a new hypothesis and go through this process again. Or now we can communicate our results. So as you saw, every experiment begins with a solid research question. It should be focused, measurable, and meaningful. Great questions often emerge from prior research or gaps in understanding. Ask yourself, what do I want to know? And why does it matter? So once you have that research question, the next step is to write your hypothesis, something that's testable. You want to clarify your hypothesis, design that experiment to test your hypothesis, and then plan your data collection carefully. So what data will you use or collect? And all of this, we want to be making design choices that are completely ethical for the people involved, for the scientific community as well. You probably heard the phrase, a hypothesis is an educated guess. The classic structure for a hypothesis is this if, then, because structure. And the reason for this is that this if then has the independent variable after it, then has the dependent variable after it, and then the because has a very clear scientific rationale. So this format ensures that your hypothesis is testable and scientifically grounded. So if we look at this question and hypothesis about CRISPR and genetically modified organisms, or GMOs, we can see how this is a testable hypothesis. So the overall question that we're looking at here is, does knocking out a specific allergen-producing gene in peanuts using CRISPR-Cas9 reduce the allergic response in sensitized individuals? So if we look at that hypothesis, we see if this specific gene is knocked out, so that's the independent variable, then the modified peanut extracts will show a statistically significant reduction in the allergenic reactivity. So the Wheeler size, smaller Wheeler size, and lower IgE binding. So this is giving us an expectation. So this then is showing us our dependent variable, something that we're going to be measuring that will show us if our independent variable or our modification in the experiment is working or not. Then our next part of the statement is because. So because the scientific rationale is that the targeted gene will knock out and prevent the synthesis of the allergenic protein, thereby diminishing the immune system's recognition and response to it. So this shows the rationale of why you would expect a smaller wheel size and lower IgE binding once that allergic response protein is knocked out. So this structure makes the logic and testability of a hypothesis very clear, connecting the intervention and the expected outcome that's measurable to the scientific rationale. So we have our hypothesis, and we want to make sure that we have the correct data sources and understanding of how to handle our critical steps in this research project. So we ask ourselves, where would you look to find data to answer your question and test your hypothesis? First places that people typically look are in these two broad avenues. So first, you can collect and conduct your own study and collect original novel data. And this gives you the maximum control over what's collected and how. Alternatively, you can explore existing data sets, often found in free repositories online. For example, many government agencies, research institutions, and academic projects make their data publicly available. One key issue to consider, especially if you're collecting your own data and using varied sources, is to make sure that you understand the different types of data that you're working with. For example, if you're working with quantitative data or numbers, discrete numbers, versus qualitative data. For example, qualitative data would be something like descriptive texts or images, videos, things that are categorical more. You want to make sure that you know how to handle those data. So specifically, if you're dealing with qualitative data, you need to make sure that you'll be able to code it systematically to identify patterns and themes. This is a crucial step in making sense of non-numerical information. So to continue this thought process and questioning what type of data needs collecting to make sure that you can test your hypothesis, we plan our experiment very carefully. So collecting data to effectively answer your research question, we have to break it down into a couple of different parts. For example, do you need different treatment group data? Most experiments involve comparing outcomes. This is usually a yes. So sometimes one treatment at one temperature versus another treatment at another temperature, all compared to, say, a control. So do you need a control group as well? As discussed, a control group provides a vital baseline for comparison. And critically, when we think about what is actually being compared between a treatment group and a control group or between treatment groups, we think about the dependent variable that you need to measure to actually test your hypothesis. So this is the outcome that you will tell in your hypothesis. And if you have that outcome met, then you know that the hypothesis is supported. Do you need a control for some independent variables to ensure that you're isolating the effect that you're interested in? And then lastly, do you need to collect categorical, discrete, continuous, or time series data to really test your hypothesis? The nature of your variables will dictate this. So answering these questions upfront helps to ensure that you gather all the necessary information for a meaningful analysis. When we design an experiment, our goal is to understand cause and effect relationships, To do this effectively, we need to clearly define and manage different types of variables. So let's break it down. First, we have independent variables, or IVs, where what we're controlling or manipulating. So these are the factors that you, the experimenter, are directly manipulating. It's what you're testing. The dependent variables, this is what we're measuring, how it will be measured accurately and reliably. So dependent variables, or DVs, is what you measure in the experiment. It's the effect or the outcome that you expect to change in response to the manipulation. Its value depends on the independent variable, the IV. The key questions here to think about are, what are you measuring to see if the IV hasn't had an effect? And very importantly, how will this be measured accurately and reliably to ensure your data is trustworthy? Say we are testing the effect of a new fertilizer on plant growth. The independent variable would be, say, the type of fertilizer or the amount of fertilizer used. You might have one group of plants getting fertilizer A and another getting fertilizer B. So we'd have multiple treatments. Then we'd also have a control, what that's getting no fertilizer. And so we're controlling the difference based on fertilizer and the amount of fertilizer. Next time, we want to think about the measurements. So in this fertilizer example, could the plant height or number of leaves or crop yield or biomass be an indicator of the amount of fertilizer and making a difference? So could those measurements be a way that we could see if our treatment is having an effect on the plant? Outside of independent variables and dependent variables, there's something called controlled variables. Now, controlled variables are also known as constants. These are the other factors in your experiment that you need to control for to keep the same or constant for all of your groups, both experimental or control. The main question is, what factors will be kept constant to ensure a fair test and to isolate the effects of the independent variables on the dependent variables? It's crucial to control these constants because if they vary, you won't know if the changes in your dependent variable are due to the independent variable or not. They could be due to these changing factors. So in our experiment here that we're using as our example, things that could be controlled variables would be things like the amount of sunlight each plant receives, or the amount of water, the type of soil, the pot size. These would be things that must be kept identical for all plants in the experiment, regardless of treatment group or control group, so that any difference in growth can be attributed to fertilizer difference, not any other factor. These are tricky factors that you might not have initially controlled for or have difficulty in controlling, but they could still influence your dependent variable and thus muddle your results. They confound your ability to say if the independent variable was caused by a change in the dependent variable. The key questions are, what other factors besides your independent variable could realistically influence the results? What steps will you take to potentially minimize their impact? For example, in our plant study here, a confounding variable might be an unexpected pest infestation that affects only some plants, or a sudden temperature fluctuation in one part of the greenhouse. So one way that some people get around this in their experimental design is that they use randomization to distribute these unknown effects to implement some type of shielding against pests, or carefully monitor and record environmental conditions. Sometimes after the fact, you can statistically control for confounding variables if you've measured them. Our experimental designs should be clear enough that another researcher should be able to reproduce our work. So all of the details, all of the steps, so the step-by-step procedure, all of the specific details, materials, conditions, precisely So when we're thinking about our experimental methods, everything should be so clear in terms of our step-by-step procedure, having clear enough specific details. For example, the materials used, the conditions for every single type of group, if randomization was used, every type of precise detail needs to be included. So other researchers could reproduce your work and get similar results. So they want to know not just what you did, but how you did it and in what order. One part of this experimental design is how we select a sample for our research, who we're studying. A sample is a smaller, manageable subgroup of individuals or items selected from a larger group, known as a population. So for some people, they study medical interventions. Some people, they study plant populations. So every study potentially has a different type of population that they're sampling from. So a sample design is super critical. And a good sample design ensures that the data we collect is truly useful, so that what makes a sample design effective is that it must be able to answer our research question and test our hypothesis. So a sample can't do that. It's not the right design. It needs to be accurately representative of the population that we're interested in. So not just the sample itself, but the population. So a good sample design is unbiased, it's ethically designed, so meaning that it doesn't systematically favor certain individuals or outcomes over others. It's also minimizes sampling error. While some error is natural, a good design keeps this at as small as possible, and it's likely close to what we would find in the whole population. Also, a good sample design incorporates time and budgetary constraints. The best design, in theory, isn't useful if it's impossible to implement. And we want it to be realistic, so to ensure findings are applicable. And then lastly, it's crucial that a good sample design considers how the data will be analyzed before we even start collecting data. This ensures that we gather what we need in the right format and in the right way. As you can see here, at the top, we have the target population that's being sampled. So that's a continuation of what we just spoke about previously. Now, when we are continuing the design of a strong experiment, there are overall 5 core elements that we need to consider. So we need to consider treatment or experimental groups. And so these are the ones that are receiving the intervention that we're studying. So for example, if we're testing the effect of CRISPR on peanut allergen response, the treatment group would receive genetically modified peanuts with the allergen gene knocked out. And then the control groups, these would serve as the baseline for comparison. So they would receive no treatment, or a placebo, or a current standard treatment. The control helps us to determine whether the observed effects are truly due to the experimental manipulation, or if they could have occurred due to other factors. Then comes replication. This means that repeating the experiment or treatment across multiple subjects or trials. So replication improves the reliability of our results by minimizing the effects of random chance or outliers. So for example, if this is our experiment, we would go through and do this experiment multiple times following this format with multiple samples. So replication is different than being able to reproduce the study. It's based on the main idea that if a result is only observed once, we can't really trust it. If it's replicated many times, we can start to gain confidence in it. To reduce bias and strengthen our design, it's really common to use randomization and blinding. Randomization refers to assigning participants or samples to treatment or control groups in a way that is completely random. This helps eliminate selection bias and balances out unknown variables, so potentially reducing those confounding variables without even knowing it. Blinding, on the other hand, prevents bias during collection and interpretation. So in a single blind study, the participant doesn't know which group they're in. In a double-blind study, neither the participant nor the researcher knows which group they're in. This is especially important in studies where expectations could influence outcomes. And then lastly, we have blocking, is a strategy for known sources of variability. Let's say we're growing plants in a greenhouse with different light zones. Blocking would group plants by light exposure and randomize treatments with each group. This way, any differences caused by light won't interfere with our analysis of the treatment effects. Together, treatment groups, control groups, replication, randomization, and blinding and blocking form the backbone of rigorous experimental design. They help us isolate the true effects of our intervention and ensure our conclusions are scientifically valid. Now, we've mentioned the definition of each experimental concept. We can focus on their purpose. So the treatment group's purpose is to test the hypothesis directly. The control group is to compare with the treatment group. For replication, it's to improve reliability and reduce error. Randomization is to eliminate selection bias. Blinding is to prevent observation bias. And blocking is to control for known variability and also potential confounding variables. Once the overall design of an experiment is decided, the next steps are to finalize how and what data is to be collected. Collecting data requires consistency and accuracy. What dependent variable you are measuring could be quantitative data as numerical, which is ideal for measuring changes, rates, and frequency. It could be qualitative data, which includes descriptions or images or participant responses. Both are valid depending on your research question and discipline. There are even ways to ask qualitative questions, but get a quantitative value. Think about when you're at a doctor and they ask you how you're feeling on a scale of 1 to 10 with a sequence of faces. Choose tools suited to your measurements and maintain consistent techniques across trials. Remember to follow ethical guidelines, especially when working with humans or animals. Once you have your data, organize it into tables or visuals to make patterns clearer. Use appropriate statistical tests to determine significance, like t-tests or ANOVA or chi-squares. The interpretation is key. What story does your data tell? Here's a helpful tool for figuring out which data visualization works best for your data. I'm not going to go through and read everything here, so this is where pausing the screen will be super helpful. The data visualization decision table is a practical reference tool designed to help researchers, educators, analysts, and students choose the most effective type of visualization based on the structure and purpose of their data. This table organizes visualization types by goals, data type, appropriate chart type or visualization, and when to use them, allowing users to quickly match their data characteristics with suitable visual formats. When we think of statistical tests, it also requires a lot of decision-making to make the right choice, because there's a lot of potential statistical tests out there, but we want to make sure that they fit your data appropriately. So here's another tool that you can use. The Statistical Analysis Decision Table is a practical reference tool designed to help researchers, educators, analysts, and students select the most appropriate statistical test or analysis based on the structure and purpose of their data. This table organizes statistical methods by analytical goal, the number and type of variables here, the types of data here, and then provides the test or analysis that would be most recommended, and then the when to use it here. Whether comparing groups, exploring relationships, or predicting outcomes, this tool simplifies the decision-making process, and it also promotes sound evidence-based analysis. So regardless of what you choose, always check your assumptions, because every statistical test or analysis type has assumption testing for things like normality, homogeneity, independence, before selecting a parameter test. If you're using a non-parameter test when assumptions are violated or with ordinal or ranking data, you want to make sure that you're using the correct test. And then just a reminder, the DV here is regarding to the dependent variable, and then IV here is referring to the independent variable. Pause is needed to check this out. Once you analyze and then visualize, you can identify patterns that are statistically significant. Conclusions that are based on your data, not your hopes. So did your results support your hypothesis? Be honest about any limitations or anomalies. Good conclusions always suggest future research directions. So really thinking about those conclusions critically. Some places where people have common pitfalls in scientific research are skipping control groups or conducting too few experimental trials. These may seem like small shortcomings, but they can be significant and undermine the reliability and validity of your results. Controls are essential for isolating variables and ensuring that your findings are truly due to the factors you're testing. Similarly, having an adequate number of trials or reproducing the study, and also increasing the statistical power of your study helps identify real patterns rather than chance outcomes. Being precise and thorough in your methodology, clearly defining your procedures so that others can replicate your work and trust your conclusions. This will help build precision and credibility. When it comes time to analyze your results, we want to be cautious. Let the data speak for itself. It's tempting to draw broad conclusions or inflate the significance of your findings, so we have something flashy or showy, but doing so can lead to misinformation or misinterpretation. Significance will be there if it's there. If it's non-significant, then it's non-significant. Scientific integrity depends on honesty, transparency, and a commitment to accuracy. These are not just best practices, they are the foundation of good science. When you uphold these principles, you contribute to a research culture that values truth, accountability, and meaningful progress. If you want to learn a little bit more, please check out these different resources. They're very helpful in helping to develop experimental designs. If you want to learn more about ethical data storytelling and how to craft a story from your data, telling that honest, true story, please check out one of our other videos.